Hello, my name is Armando Salais.
I'm Erica Garcia.
My name is Sergio Marquez.
My name is Jessica Ramos.
And my name is Darian Ramos.
And today we're going to be talking about membranes. So we are going to do an overview of what the presentation is about today. Our project that we got assigned was about membranes. And so today we're going to define what a cell membrane is and describe the function. We're going to provide a brief explanation of the fluid mosaic model and explain how fluctuating temperatures affect the fluidity of the cell membrane. We're going to also go over the results of the investigative work and give perception on broader impacts that it has. So as you can see, we have a picture here kind of a membrane. And the membrane is a pliable sheet of tissue that covers the lines or connects the organs of cells and animals and plants. They are mostly constructed of a lipid bilayer. The lipid bilayer consists of 2 layers of glycolipid, cholesterol, and phospholipid molecules. They determine if the membrane is formed in round vesicles or flat long sheets. So the cell membrane is a thin semi-permeable membrane that surrounds the cytoplasm of a cell. All living cells contain a cell membrane, and the semi-permeable structure is kind of what surrounds the cell, and it allows certain substances to pass through or not to pass through. And that's some of the things that we'll be explaining about later in its function. This is just kind of an overview of what a membrane is.
So the function of a membrane gives support and balance to a cell, and they keep toxic substances from reaching the cell. They serve as a gate for other necessary things to be able to pass through the cell, such as nutrients, ions, metabolic products, and waste. And they separate important but also not compatible processes within the organelles. And the next slide, please. So here we have a molecular view of the cell membrane at the top. And at the bottom, we have the fluid mosaic model, which I will briefly go over. So the fluid mosaic model tells us about the cell membrane, and it tells us if it is fluid. Phospholipids, which you could see the phospholipid is the light blue circle with the black looking strings. Those are the tails. Phospholipids are free to move in any direction. So the cell membrane is composed of proteins, phospholipids, glycoproteins, glycolipids, and cholesterol. Today, I will only be going over phospholipids and cholesterol because they are the 2 most important components of the cell membrane. So the cell membrane, as said previously, is semi-permeable, which allows things to go in and out of the cell. And 2 examples of that would be oxygen and carbon dioxide. The reason for that is because they are small nonpolar molecules and they can easily be diffused in and out of the cell. So I'm going to discuss the phospholipid. The phospholipid has a polar head, which means that it's hydrophilic, meaning that it loves water, and they can be seen at the top and bottom portion, right next to the peripheral protein in purple. And then the non-polar tails are going to be facing inside the membrane because they do not like water, which is also known as hydrophobic. And then I'm going to go over cholesterol. Cholesterol acts as a buffer and it maintains fluidity of the membrane. So when you increase the temperature of a cellular membrane, the phospholipids will move apart from each other. And that means that the cell becomes more fluid. Cholesterol will try to decrease fluidity when that happens, so that it will prevent the cell from dissolving. When fluidity goes up, cholesterol tries to bring it down and vice versa. So in the following slides, we will see how playing with temperature will affect the fluidity of the cell membrane. So our question and hypothesis. Our question that we asked is, how does temperature affect the cell membrane? Our hypothesis is if we lower the temperature, the cells will probably lose mobility and their growth will stop because the phospholipid bilayer becomes inflexible.
Our predictions for this experiment was that with low temperatures, membrane cells will have problems to survive and the membrane will not allow any molecules to pass through, and that at high temperatures, the phospholipid bilayer will dissolve and also become more flexible, which will allow unseen molecules to enter. The methods we used for this project was working with the fluid mosaic model, which we were able to determine that by rising the heat, the membrane is more permeable. By moving around some of the buttons on our virtual lab, we were able to see that the permeability was affected either more or less depending on what factors you moved. And the temperature was measured from a scale from 1 to 10. And on the bottom, you can see the link to our virtual lab. The independent variable for the methods that we use was changing the temperature from scale 1 to 10 on the fluid mosaic model.
For the dependent variable, we keep the numbers of atoms at 200 because if we change the temperature, the atoms stay at 200. And the waiting on results around 1000 clicks in the fluid mosaic model for the same reason, because if we change temperature, the 1000 clicks stay the same. For the results, we conclude that if we change the temperature, we can clearly see that the membrane is more permeable faster than leaving it at 1. Based on the stimulation, the phospholipids in the membrane is spread as the heat temperature is increasing. In the picture number 1, we can notice that all the molecules stayed together and at the same place because the temperature is lower. And in the second picture, all the molecules are around the membrane because the temperature is increasing. For the observation, we noticed that the high temperature increases the fluidity of the membrane. The best temperature of the cells to function is above 98.6 degrees Fahrenheit. When the temperature around the membrane increases, this causes more fluidity. The problem with more permeability on a membrane is that it can allow harmful molecules to enter the cells, and also decreasing the temperature will not allow the fatty acids tails of the phospholipids move around and they become hard or inflexible.
So in conclusion, we learned that temperature is crucial in the cell membrane, and that is also a factor that alters the components. The temperature needs to be 98.6 degrees Fahrenheit in order for everything to flow properly. The membrane is permeable and does not allow any particles to pass through. So the body impacts is when temperature rises, it can cause proteins to start breaking down. Decreasing the temperature also affects cells negatively by preventing the cell to increase in size. One way that you can potentially kill a cell is by decreasing the temperature. The result of this project is to inform society that cells must be permeable to some extent to be able to perform homeostasis. And this is the sources that were used in our investigation.